Welcome. I'm Liz Taylor, ocean explorer, naturalist, and entrepreneur.
I'm Sylvia Earle. I'm an oceanographer, National Geographic explorer, founder of Mission Blue, and I'm an ocean elder. Thank you, ocean elders, for sponsoring this program.
Yes, and this is our show Dive In, where we host informal and open conversations with the ocean community on topics of wonder and interest. We're glad to be back, and we hope everyone has made some time to get outside.
Yes.
You can put your questions into the Q&A box, and we'll get to as many as we can towards the end of the program. And before we get underway, we want to remind everyone that the world
Is
blue.
Check it out.
Yes. So today we are so excited to be speaking with Professor Andrew Sweetman.
Yes.
Here he is. Professor Sweetman leads the seafloor ecology and biogeochemistry research group at the Scottish Association for Marine Science in the UK, and chairs the benthic ecology and biogeochemistry department. He has a PhD in deep sea ecology from the Max Planck Institute for Marine Microbiology and has held research positions at various institutions, including Woods Hole, Florida State, University of Maine, and University of Hawaii, and the University of Bergen. Welcome, Andrew.
Thank you very much. Lovely to be here.
So good to have you.
You have the qualifications of a very nerdy scientist. But you also get out there.
I don't have any letters after my name.
Wash around in the real ocean.
Yes, he does, with all kinds of amazing tools and sensors and techniques to really dive into it.
And thank you.
Think about ocean explorers on the Challenger expedition in the late 1800s, what they would say if they could come and see what is now known.
Mm-hmm.
We still don't know very much.
No.
A lot more because we have technology that didn't exist. And now.
Yeah, it's but we've still explored so little. And in terms of deep sea science, we tend to go straight to the seafloor. Most of the time. The largest deep sea habitat, the largest habitat on the planet is the water column.
That's
and the
right.
deep sea
That's
water column.
Ocean,
it's
between the top and bottom.
We typically, when we go out to sea, we're with an ROV or a submersible or whatever, we're straight down to the bottom. The cameras are on, but nothing's being recorded.
Mm-hmm.
I often say to researchers or the ROV subpilots, just record everything as you're going down. You might see something interesting, but
you might. She always says that's the juicy bit, right?
It's true.
Yeah.
But most of your research is kind of focused on the benthic part, the very inaccessible, challenging pieces. How did you get interested in working in the seafloor region?
Well, I mean, that's how long's your program? For me, it all started. I grew up, in the 80s in the UK. My parents were moving around all the time. So I think I went to about 13 different schools by the time I was 12. So, I'm sorry?
That is an education in itself.
Well, it's an education, or it's a lack of an education, because I would leave one school when they were just about to start a topic, and I would come into another school where they just finished that topic. So there was a lot of things that I missed, and eventually at 12 years old, when you can't really read or write and add or subtract, it was a bit of a tough period. So I was drifting like the plankton, I guess, through school. And then one day I went to the library, and there was a book by Robert Ballard, The Discovery of the Titanic. And I picked it up because my great grandfather, he helped build the boilers for the Titanic. So I
cool.
was a bit drawn to that book. And then I took it home one evening, and I was looking at it, and in the morning room, and my mum came in, and she was asking me what I was reading, and I turned over to this picture of this in the book, Woods Hole Oceanographic Institution, and for some reason it just jumped out at me, and suddenly I realized that I wanted to work there, and so the next day I started applying myself and learning to read, learning to write, going to extra math classes and everything. And 8 years later, I was working there. So
Wow,
I,
incredible.
yeah, and it was I started working in a seafloor ecology lab that was run by Dr. Lauren Molyneux, an amazing deep sea benthic ecologist, seafloor ecologist, and yeah, I just fell in love with the deep sea and deep sea biology and the seafloor environment there,
Yeah.
and been with me kind of ever since.
This is
We,
the power of books.
it is the power of books and the power of library,
Yeah. Well,
having accessibility.
well, I couldn't read much of the book at the time, but it was the power of the pictures.
Yeah, yeah,
And
it's the photographs really tell the story and a
yeah, yeah,
history.
yeah, yeah.
But you spent quite a lot of time out there in the deep sea at this point. And what are some of the really interesting things that you've come across?
Like dark oxygen.
Uh,
Like dark oxygen,
well,
yeah.
it's such an amazing environment. What I love about it is that whenever you go down with an ROV or a submersible, or you send a lander down, sometimes with cameras on it, and you see the ocean floor through an image or whatever, you're the first person in the
Yeah,
history of our species often to have seen that, you know.
Hmm.
that's right.
I mean, some sites, for example, the Pacific Rise, it's quite well studied. So when you go down in a sub there, you go into sites, locations that have been visited by a few people, but in the abyss, it's really
Mhm.
a black hole in terms of knowledge relative to other types of habitats.
Yeah.
And you know, every every you know rock that you see, every fish that you see, you're the first human
Yeah,
eyes in the history of our species to
and vice
see that.
versa. You are
Yeah,
the
yeah,
first primate they have ever seen.
Or
yeah,
the first
yeah,
robot.
yeah. Yeah, I remember diving in the Alvin. It would have been 2019, and I've done a lot of work on deep sea fishes. And when we bring them up to the surface, they often have quite significant barotrauma, and they die.
Mhm. That's
And
so
they,
sad.
it's just one of the issues with bringing up deep sea animals. A lot of them just can't survive the pressure decrease. But so I'd seen them in pictures, and I'd seen them, on the deck in our baited traps. But then when I got down there and there was this rat tail just swimming in front of the viewport to the Alvin, and I was like, "Oh, that's what you look like." You know, it's I think it really calibrates your gut, to go into
Mhm.
the deep ocean and go down and
Directly exploring.
yeah,
Yeah.
and
I
and
often
look up
think,
at the hatch and realize there's 2 miles of water above that hatch, and it's
Yeah.
think about what
it's
we
not for
know.
everyone. My
Yeah,
my my father, he would never do it, um, but I love it. I can't get enough of it.
That's excellent.
Yeah,
what
yeah.
would we know about anything on the land if we had to be explorers by using standard oceanographic techniques?
It'd be pretty sad.
I
If
think
we
about
Yeah,
didn't.
Jane
yeah.
Goodall getting to know chimpanzees
Hmm.
on an airplane.
Well,
Yeah.
it's I think the other interesting thing when you think about it, and I heard someone say this quite recently, and I thought that's a really good way of, of thinking about it. But you know, if there was an alien that came and they just dropped a probe down to Earth, it would most likely fall into the deep ocean because
Yeah,
the deep
Correct.
is most of our planet. And if it went down all the way to the seafloor, it would most likely collect a sediment sample.
Hmm.
And within those sediments, you'd get little microbes, you get little nematodes, forams, polychaetes, annelid worms, things like that. And then
mollusk.
if yeah, and then if it pulled that sample back and it looked at it, it would think that the planet Earth was composed of microbes, worms, and mollusks, and it would miss everything else.
Miss everything else.
And
Incredible.
it's often the same way of thinking about it when we think about land, there's a hidden world out there. In the
Hmm.
ocean and in the deep ocean, it's even more
Yeah, but think of
more
the
nebulous.
privilege that we are the
I'm sorry.
first, as you
The
point out.
privilege.
It
Mhm.
is. How humans have been around more or less for 300,000 years. Of course, sharks have been around for 400 million years.
Mhm.
And we're the first ones to have technology, even to go scuba diving. I mean, the
Mhm.
last 100 years, and especially the last 50 years. And it just keeps getting better and better in some ways, being able to sense and understand the world around us at the same time that perversely we are destroying the very systems that keep us alive. It's like a
Yeah,
race. Do we
yeah,
move enough fast enough?
yeah, it's really really worrying. And when you I mean, I teach a number of classes at SAMS in Western Scotland, and often a lot of them, whether you're talking about oil and gas extraction or climate change,
Hmm.
a lot of the students get quite depressed.
Yeah.
And but I often say that the first step in solving a problem is realizing there is one.
Yeah.
And
100%.
yeah, and
And
it's once you know that, okay, we got to do something about this, then you can modify things, processes, procedures to limit the damage as much as possible. Um, and sometimes, for example, with marine protected zones, marine protected areas,
hmm
eliminate it altogether.
yeah.
Climate change is a bit more of an issue to deal with, but
Yeah. It was interesting the work that you did with or are, have done and are doing around just the jellyfish in the sea, right? And the role that they play in the whole
yeah,
carbon
yeah,
pump cycle.
yeah. I mean, that was a bit of a strange one. When I was growing up, I used to annoy my school teachers quite a lot because they would, you know, tell us about biology or tell us about chemistry, and I would always be asking why. They'd say, "Well, this happens because of this," and I said, "Well,
As you
well,
should.
why? Why does it happen like that? Why can't it happen another way?" And I used to drive my professors, my teachers potty. But they put up with it, and when right after I was working at the University of Hawaii, I was living in Norway, and we had a lot of problems with jellyfish in a lot of the fjords because of a lot of the aquaculture was putting out all these nutrients into the water
Hmm.
column. There was a lot of coastal water darkening,
Mm.
and it's still going on. And fish, they're visual predators. They need to see their prey to catch them. Well, jellyfish, they just drift around with their tentacles out. They're
Mhm.
they're tactile predators, so they can um, compete a lot better in a dark water column compared to fish.
Wow.
And when the jellyfish start taking off, they start competing with the fish for food, but they also start eating the small fish. So once you get a jellyfish dominated ecosystem, it's very difficult to get it back. And I started looking into this and learning a lot about jellyfish blooms, what causes them, what are the impacts of them. And then I would say to myself, well, okay, a lot of these jellyfish, they have seasonal life cycles. They go from the fiery stage, the larval stage, to a jellyfish,
Hmm.
and then they die within a single year. And often they die en masse. And I was often asking myself, well, what happens? Where do they go?
Yeah.
And there was no research that was being done on this. And primarily it was because people have thought that, it's 98% water. It's just a dead end in the food web. Nothing's going to eat it. Um, and then that question came by, well, why? A question that I annoyed my biology high school teachers with. I said, well, why won't anything eat it? So I wrote this grant proposal. We got about £500,000 to go out and study it. And I brought some brilliant scientists along with me, Craig Smith from the University of
Oh
Hawaii,
yeah.
Fabio De Leo from Ocean Networks Canada, Dan Jones from the National Oceanography Centre in the UK. And we went out with these landers, and we made them exactly the same. So there was no difference in the artificial reef effect. And we put dead fish down first. So we deployed dead mackerel, put it down on the ocean floor, and 24 hours later, we recovered the landers from 1,200 meters. A lander is basically an instrument that you put into the water, you release it, it sinks by itself, does its thing. Then you come over with a boat, you tell it to release its weight, it releases the weight, and all of a sudden it's positively buoyant and floats back to the surface.
Mhm.
Um, so 24 hours later, we came back, pinged it, it came up, and there was nothing left of the fish other than bones.
Wow.
And there was loads and loads of these hagfish in the images. And then we started to think, okay, let's put down the jellyfish. So we put them out in similar ways on the landers, put them down, and we didn't think we were going to see much because I mean, even myself was thinking that that there's probably not that much that's going to come and eat them. We never see them on the sea floor, so they're probably not coming down that much. Um, and we send the lander down, and 24 hours later, um, the lander comes up, and the jellyfish are not there. And then Craig and Fabio and Dan and myself spent about 4 hours trying to redesign the lander because obviously the jellyfish had washed off on the
Ah,
way home. And then I decided, well, I now need to download the pictures. And I downloaded them and I realized that the jellyfish had arrived intact at the seafloor. But they were gone within an hour because the amount of animals that came in and consumed
wow.
them was incredible. We had just as high a biodiversity of scavengers consuming the jellyfish as the fish. And suddenly it dawned on us that the reason why we weren't seeing any of these jellyfish on the seafloor underneath these blooms was not because they weren't coming down. It was that they were coming down in such a high quantity but the scavengers were just removing them. So when you came along the seafloor with an ROV, you just didn't see them. So
Incredible.
it really changed the way we think about jellyfish in deep sea food webs. And it was all because we kind of said, well, let's go out and test it. Let's go out
Yeah,
and
good to
yeah,
see.
because no one's done it before. And yeah, and then and since then we've been out many times and found the same thing in different areas. And not only that, I had a brilliant postdoc, Kathy Dunlop, who's now at the Institute for Marine Research in Norway. She did an experiment, and we actually found commercially exploitable species consuming dead jellyfish.
Wow.
And she found that, that the the jellyfish populations in some of these fjords could actually support some commercial fisheries, which was pretty incredible.
That's pretty wild. Yeah, everybody's
Yeah.
just like, oh, a jellyfish, but they do have a role. It's amazing.
Yeah.
Well, they also have a role in the twilight zone.
Yeah,
Some
yeah.
creatures dive down, certainly leatherback sea
Sea
turtles,
turtles, yeah.
Hmm. Yeah, yeah. And
and
in the
those
deep
inside.
ocean, I mean, the biodiversity of deep water jellies is pretty incredible.
Yeah.
Yeah.
But I guess the most controversial topic in ocean science, I think, is the whole thing around the deep sea mining that you've been so thoroughly deeply involved in,
So to
and
speak.
so
Yeah,
to
yeah,
speak.
since 2013.
Yeah,
Yeah.
and that's this idea of just removing the nodules and crusts from the ocean floor, and industry tends to really characterize the seabed that we've been talking about as being so rich in biodiversity. They call it a biological desert, devoid of life, covered with rocks that are just there for the taking. And it seems
Mm.
like nothing could be really further from the truth. Um,
No, I mean the one of the things that's often says is that they're low biomass habitats, and then that that's true. They are, generally. You do find very, very low biomass in some of these habitats. The abyssal plains, for example, very, very high biodiversity, but very, very low biomass. Um, but there are lots of other low biomass ecosystems on planet Earth that are critical to, our survival. For example, the Arctic sea ice,
Yeah.
okay,
Yeah.
which reflects sunlight or sun energy back into space and keeps the planet warm. That's a low biomass habitat. If we remove that, if we lost that, that's a pretty significant thing. And another perfect example is the Galapagos Islands. That's a low
Mhm.
biomass habitat, relative. You don't find huge forests there, um, yet the the discoveries of Darwin that laid the foundations for the theory of evolution, which has led to amazing breakthroughs in, medical research and, biology and, you know, survival of the fittest. It's probably impacted, financial models as well, and, economics. That all came from a low biomass ecosystem. So
Yeah.
just because something is low biomass doesn't mean it's low impact. Uh,
Exactly.
you
Or
know.
the way we tend to measure biodiversity, we're talking about the splintery ends of diversity species.
Hmm.
And we've spent all of our history getting to know creatures who live on the land,
Hmm.
and especially insects are so diverse.
Yeah, yeah.
It appears that biodiversity is terrestrially biased, that more species, more than double, that have been identified from the land, it's only 3% of the biosphere.
Hmm.
And that the impression that the ocean is a dark, deep, bleak place,
Hmm.
Yeah.
except for maybe coral reefs and kelp forests.
Hmm.
But they're up here, and most of the ocean is deep and dark and cold.
Yeah, 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 yeah. It's
And
it's it's
and
true,
it's
but
not species, it's you know, all the major divisions of animal life
it's
are in the
it's genetic
ocean.
diversity, it's food web diversity, it's habitat diversity,
Yes.
it's all biodiversity. Um, and I think the one thing that uh, is is interesting is that in the deep ocean, with the exception of, six-gill sharks and, giant squid and things like that, and, some deep diving whales, you don't get that much charismatic fauna down there,
That's true.
other than,
Depends on the
deep
definition.
sea
That's
fish
true.
every once in a while. It's normally really tiny and you need to look at it under a microscope, but the functions that these ecosystems they carry out, they're immensely important. And I often try to teach my students about the importance of biodiversity, but also the importance of function in ecosystem.
Hmm.
And I often use the example of, imagine you're in a restaurant and there's lots of people eating and walking, and people are cooking and people are taking orders, people are going to the bathroom, people are talking. Um, Within the same restaurant, there are, hopefully, if it's a cleanish restaurant, there aren't that many, but you'll get spiders, you'll get ants,
Yeah.
you'll get other insects, you'll get bacteria. So your biodiversity is quite high. Now, if you were to take, for example, all the ants away, you've reduced your species richness, the number of species within that restaurant ecosystem by one, but your function of the system hasn't really changed that much. Now put those ants back in and then take away the people. Again, your biodiversity has gone down by one, but the function has dramatically changed.
Yeah.
Yeah.
And, that's why it's so important to not just look at what lives there and how many organisms live there, and not just focus on the animals, but also think about the microbes too. And in the, you know,
Critical. Yeah.
yeah, critical. And they make up probably most of the biodiversity and most of the biomass in some deep sea habitats.
Well, I think one of the most interesting things that you've been working on is the role that the deep sea has in producing oxygen, right? We haven't really ever thought about that even being a thing. We always think we have to have like sunlight and plants for photosynthesis to produce oxygen
Yeah,
back
yeah,
into the environment.
But chemosynthesis has been well known,
Yeah,
but it
yeah,
can proceed without the kind of environment that we take for granted.
but it was
yeah,
in
yeah.
but it was in I guess was it in the work in 2013 in Hawaii? I think we were part of our team was out there with you when you deployed your landers and you brought up the samples that
Yeah, that was 2018.
or
Yeah,
20 yeah.
Ian
But
was out with us in
yeah,
yeah.
but that's where you came up with some of the samples that had bubbles inside that you didn't expect to find.
No, that it happened in 2013.
13, yeah.
Yeah, the whole story. I was talking to a friend of mine about this yesterday, and she might be online and she's probably giggling now, but the whole story about how this came about is very, very bizarre. Um, because my whole background doing my PhD was doing flux measurements, measuring the the amount of oxygen consumption and the amount of CO2 production and the amount of nutrient cycling that goes on in the seafloor. And for that, we use these a variety of different instruments. We sometimes use microsensors, other times we'll use seafloor chambers. And just to give you a view as what an idea of what a seafloor chamber is, it's basically just a bell that you put over the seafloor, you
Hmm.
stir it, and you measure the oxygen concentration continuously. And just as if I were to, someone were to put a big bell over me right now and tape all the air gaps and stick an oxygen sensor in, as I was breathing and metabolizing, the oxygen concentration would go down. And that's what
Right.
we were doing, or that's what we were trying to do. And we've been using these instruments, many different types of landers for years, deploying them in the Arctic Ocean, the Atlantic, and uh, other parts of the Pacific Ocean, and just measuring this seafloor respiration, this oxygen consumption. We call it SCOC, sediment community oxygen consumption, because
Hmm.
scientists always need these
Acronym.
terms. You know, you know.
Did you want to put this, did you want to share your slides a bit to
Yeah,
walk us
yeah,
through this?
I can share some slides. Sorry, I forgot all about it.
Yeah.
Yeah, so yeah, can you see? Can you
Yeah.
see this?
You might just
Yeah,
go to
yeah,
slideshows
yeah,
up there at the top. Yeah.
yeah.
Here we go.
Continue to share. Yeah,
Yeah.
yeah. So this is just starting to talk about where the ocean gets its oxygen, the deep ocean gets its oxygen from, and it's largely from, and I still think it is from deep water circulation,
Hmm.
photosynthesis in the upper ocean, and on land it produces lots of oxygen, and at the poles where the water is very, very dense, it sinks into the deep sea with lots of oxygen, and then it circulates around the world's
Right.
oceans. That provides the oxygen for aerobic life to exist. And you can measure this biological activity by measuring the rate of oxygen consumption at the seafloor. And these are the instruments that we've been building and redeveloping, and building new ones over the last 15, 20 years. These are what are called seafloor respirometers, or benthic
Cool.
chamber landers, and they sink. That we put them in the ocean. Um, they sink autonomously by themselves. We release them. They sink to the seafloor, and then they carry out an experiment. And then we come over their location and we send a signal down to them to release the weights, which are on these legs, these
Hmm.
iron-like looking things. And then that releases the weight, and these big orange floats bring the whole system up to the surface. And during the experiment, what we see all the time in other areas where we deploy these is, over time in these chambers, these white chambers, the oxygen goes down, and then you can calculate an oxygen consumption rate just from the change in concentration through time.
Hmm.
Um, and these instruments, they've been used for over 30 years, been deployed hundreds of times, and we've done it many, many times as well, and every single time we deploy them. In other places, this is generally what we see, and this is some examples from the North Atlantic or the Central Atlantic that a former student of mine published, and you can see that the oxidant just goes down slowly over time.
Right.
And I'll just stop sharing the slides for a minute. I'll keep on talking, but yeah, we were doing these experiments in 2013, and the lander came up, and in the syringe samples that are collected by the lander from the chambers through time, they were just bubbling. It was like a can of soda that was just
Wow.
fizzing, and we just didn't really. And there were these big bubbles in the syringes, and we were like, well, where's that coming from?
Right.
And Craig Smith, great friend of mine and brilliant oceanographer, and I were just scratching our heads because we'd never seen anything like this before. He deployed similar landers with others and had never seen it. I deployed landers like it, had never seen it. And so we went back to shore. We sent the components back for testing. We did more tests on them in the lab, found that they weren't, trapping air, things like that. Got the sensors recalibrated and tested. And then we went out in 2015. And again, we saw the same thing. The oxygen was just going up. It wasn't going down. We just couldn't measure
Does not compute.
oxygen consumption. It was just like, this is so weird. Why does the equipment work everywhere else when I take it everywhere else? But then when I'm here, it just doesn't work. I'm
And
not
that was
doing it
all,
different.
is that all in the Clarion-Clipperton zone?
Yeah.
All these experiments.
Yeah. And then in 2018, we went back, and again, we took a different instrument this time, a brand new instrument with different oxygen sensors, and saw the same thing again. And by this point, I was getting very, very annoyed with the instrument. I was getting very annoyed with myself and just trying to figure out what I was doing wrong, but I couldn't quite figure it out. And I contacted the oxygen sensor company up, and I said, are you sure you haven't crossed the wires so that the oxygen sensor shows an increase rather than a decrease? Thinking something like that. And they were like, no, everything's working fine. They calibrated them again and they sent them back. And then we'd see the same thing. And then in 2021, we went out, and I decided at this point, I just didn't trust the oxygen sensors, and I didn't trust the instruments. So I took out another way to measure the oxygen consumption. And we sent the lander down. The lander sat on the seafloor for 48 hours. Well, no, I think it sat on the seafloor for about 3 days because we had this ventilation period
Oh, nice.
for about a day and a half. And then the system came back, and in the ventilation period, which lasted a day and a half, you can see that the oxygen in the chamber just doesn't change. It just actually decreases rather than increases. And then the moment the chamber was pushed into the sediments, the oxygen starts going up.
Wow.
And and I was overlooking my student's shoulder who was downloading the data, and I saw this. She had this line on her screen, and it showed something going down, and I thought, yes, finally, we got consumption of oxygen. And she said, no, Andrew, that's the light signal. And the light signal works inversely to oxygen concentration. So if it goes down, it means the oxygen's going up. And at this point, I turned to her and said, right, I want you to take those oxygen sensors, and they cost about 10,000 pounds each, and
Wow.
put them in the bin because these things just do not work.
You
And then
don't
I
just
went
buy
to
that
the
off the
other
shelf.
No.
method of measuring oxygen, which was a chemical titration. And the first sample that we titrated, excuse me, an oxygen concentration bang on where we needed it. It was around 160 micromoles per
Hmm.
liter of oxygen, 165, something like that. Um, and then the next one was like 180, and then it
Wow.
went up and up and up and up.
Up, up, up.
And suddenly we realized that these two different methods of measuring oxygen were showing the same thing, that the oxygen was just going up. And these are the
And there they
nodules.
are.
Look at
These are
that.
the nodules at the seafloor. This is what they are like. They're actually little places where there doesn't seem to be any nodules, but they're actually there. It's just they're covered in sediment.
Huh,
So they're covering the entire seafloor. Yeah, and this is what we were seeing. The oxygen was just going up.
that's wild.
Uh, and then we just went through a series of different experiments to try to figure out if we were doing anything wrong, and different calculations, really. Were we dragging a bubble down? Were we, was there oxygen leaking from the plastics?
Anything that could make it make
And
sense.
everything, when you put it all together, the chances that all these contamination sources was just so minimal that we came away and thought, well, there must be something going on there. And now other people are finding it now in the labs as well around
That's
the world.
excellent.
So they're emailing me saying, yeah, we're seeing oxygen generation in the presence of manganese oxide. So we're still we still don't really know the cause. When we published the paper, we put down several hypotheses
Hmm.
about what we thought could be happening. We've and I can go through some of these with you if you want, or
Now we're curious. We want to know, like, why do we think this is happening? Is it,
well,
The metals.
I mean, I'll just go to the next slide here. We did some experiments where we just put in nodules into manganese oxide rocks, into a chamber. And this is what it looked like about 24, 48 hours. I can't actually
little
remember.
bubbles,
Yeah,
big
there's all these
bubbles?
bubbles, but they weren't there before. And these nodules, they were never dried or anything. If you dry them and put them into some water or something, they start bubbling, but it's just air coming out of all the pore spaces. So you
Right.
keep them hydrated. And we incubated them in the dark for 24, 48 hours, however long it was, and underwater. So there was a seal. The cores were sealed, and then they were under a water seal.
Hmm.
And if I just play this,
Oh yeah, let's see.
and I'm just going to just lift up the core and bang it down. So these nodules have never been exposed to any air. You can see that there's no way for air to get in through the bottom. We're just going to see if there's any microbial in the nodules. And it was all
Oh,
this
look at
stuff.
that.
Yeah, it was just coming out. So
Those little
we
tiny
think that there's
bubbles.
Tiny bubble.
yeah. And then this is the oxygen data from that core, and there is a control that we did at the same time, and you can see the initial increase in oxygen for the first hour of the experiment, and notice that the decrease in oxygen in the one with the nodule is less than the decrease
Mhm.
when there isn't any nodules. It's almost like the oxygen's been buoyed up by
Yeah.
the presence of these manganese oxides. But yeah, we have all of the oxygen. We did a number of experiments, and what we eventually found on the ship, we tried to poison some cores to actually reduce the microbial activity to see if it was a biological process or an abiological process. But the only thing that we had on board was mercury chloride, and
Oh,
there was a recent paper that just came out late last year that said most microbes in the CCZ have the ability to detoxify mercury chloride. So,
they're always getting
even
up
though we
one over
tried
on
to
you.
kill everything, it's possible that even in those experiments, there were some microbes that were still alive. Um, but in some experiments, we just incubated nodules, and there we found, we found there was oxygen production. We did a variety of different tests. We checked if there was oxygen leaking from the plastics, if there was oxygen seeping from the sediments. And then, yeah, we got to the point where we couldn't explain about 80 to 99% of the oxygen, and we ascribed that to this unknown process that we think is in situ oxygen production.
And if you think
We-
about it, there's these pretty vast areas out there that are covered with these nodules. That's quite a bit of oxygen contribution as it's going to rise up through the water column, right, to the
yeah,
surface.
I mean, I'm not so sure that it has that big of an effect on the oxygen budget there because that environment is so well oxygenated anyway.
Hmm.
It gets so much oxygen from thermohaline circulation that we don't know if they're producing, these environments are producing oxygen continuously or it's just a transient thing that maybe we stimulated when the lander came down and disturbed the seafloor. It's
Hmm.
possible we kind of like turned on a switch and then it generates oxygen for 12, 24, 48 hours and then stops. So these are all big open questions that we have. But what we were able to see in the sediments is that there were definitely microbes. About 30% of the microbes that we were finding in the sediments were a particular kind that is able to generate oxygen in the dark. So there could be a microbial role.
Hmm.
But we also wanted to explore whether the nodules were having an effect because there's all these metals in them, and we were thinking, could there be like a battery effect, some sort of splitting of water into oxygen, and so we started probing them with um with voltage meters, and we found quite high voltages on the surface of these things.
That's really interesting.
Anytime you get a resource with different metals at different oxidation states, you'll get a voltage difference. It
Hmm.
doesn't mean that there's going to be a current flow. You need current flow as well to not just a voltage to get water splitting. But we found very high voltages, um, and we only measured about 150 points on 12 nodules,
Wow,
but we
this is a
found
lot.
up to So, well, it's a lot. It took a long time, but what we hypothesized in the paper, which a lot of people, well, not a lot of people, but some people took didn't I I don't think they properly read the manuscript. What we were trying to say was that we did 150 measurements on 12 nodules total. So on each nodule we did about 20.
Hmm.
Um, or 10 to 20. And on those 12 nodules, we found a voltage that was 0.95 volts. And then we said, well, it's possible that had we done 10,000 measurements on 1,000 nodules, we could have found the actual voltage that you need.
Yeah.
Um, but then people said, well, we were challenging the second law of thermodynamics, and that we were like, well, that's not what we were saying. We were saying that we found high voltages and there could be even higher ones.
Yeah,
Um, you
exactly.
know,
You have to
we just
have
need
more.
to probe. But what
Yeah,
we
more.
we I can just go through because I just want to just go through here because this is the most interesting part. Um, because you were talking about the causes. Um, so we found these very, very high voltages. But the interesting thing, and I'm not an electrochemist, but what I've learned in my meager amount of research into electrochemistry uh, is that there can be sort of like a poisoning effect of chlorine in seawater against
Hmm.
oxygen evolution. But when you have nodules, that poisoning basically just disappears. So
Interesting.
and this graph actually shows it. The poisoning is from chlorine being released during as a current goes through seawater. Electric current goes through seawater, and that normally poisons this oxygen evolution reaction. But when you have nodules present, you can see that the amount of chlorine being released is much, much less. So they actually buffer it. They stop that reaction from taking
It's
place.
so interesting.
Yeah, yeah. They're amazing, amazing deposits. So we have this paper now that it's currently in revision. Um, but what we were able to do was do some, you know, we did some Monte Carlo simulations to see if we could actually get, um, it's a type of computer simulation to see if we
Uh-huh.
could, is there a way that we could get oxygen being released in the presence of manganese oxides at the voltages that we measured? And amazingly, we found that our green line here is at 0.952 volts, and the left peak is the voltages that you need if there's current to generate um, oxygen from water.
Hmm.
And and we found that yeah, simulations suggest that even at 0.95, you could get parts of the electrochemical splitting of water actually happening.
Very
So
cool.
so
So,
that's really
mm,
interesting. We got to figure out well what's on the other side of the equation, figure out where the protons and the electrons go. But um, and then a colleague of mine in New York, she emailed me and she said, "Oh yeah, we found this in the lab. We went out to lunch one time and we left some manganese oxides in some electrolyte like seawater in an anoxic fume hood, and we had an oxygen sensor there to so that we could make sure that it stayed anoxic. And then we came back from lunch and the oxygen alarm was going off because
Yeah,
oxygen. So
that's great.
um, so yeah, some people are starting to actually see this, and there's other papers now coming out saying that nodules are really, really good catalysts for water splitting. Um, and then we started probing whether we looked at the microbes in the nodules and the sediments, and all these boxes are around microbes and that are actually known oxygen producing
It's
uh,
incredible. So
uh,
I think leaving them
microbes.
alone on the seabed seems like a really good idea.
Yeah, it's I think I'm if we're talking about mining, I mean I'm I kind of like sit on the fence with deep sea mining. I I'm really, really worried about climate change affecting the deep ocean because that will be a global,
Hmm.
impact.
Yeah.
And I think that there may need to be maybe some sort of mining, but if that mining is going to go ahead, then we need to do it in the right way. We need to get as much information as we can about the ecosystem before we go in and do it. And
Andrew, these
yeah,
systems,
I'm sorry, Sylvia.
these systems have been developing long before there were humans.
Yeah, yeah,
And the world we came into was the result of hundreds of millions of years
yeah, yeah.
of the kind of processes that we're just beginning to understand. And you
hmm
are in the thick of discovering
Yeah,
some of these
yeah, yeah. It's
processes that we just have not known about.
Hmm.
In fact, it wasn't that long ago that no one, for hundreds of thousands of years, our history goes back maybe 300,000 years, that
mhm,
nobody knew what air was made of.
That's true.
We had
mhm.
no idea about
Yeah,
what
yeah.
oxygen is, let alone how it is produced. So
Yeah.
we're
The-
still at this cutting edge of figuring out how did we get to where we are and what are we doing to disrupt these processes that move at time scales that are just beyond our thinking. We live
mhm,
within about a century as individuals in our history as a species, given, say, the history of jellyfish, the
mhm,
history of sharks, or the history of manganese nodules that are not just dead rocks, they're living systems just
mhm,
powered by microbes. I
mhm.
mean, it seems we should show some respect and not
Yeah,
just brush
I
in.
think the more research that we're doing on in the deep ocean, and we're finding that the recovery dynamics are just from any disturbance,
Hmm.
are. are infinitely long. And even from small scale disturbance experiments, we've been involved in some mining experiments where the seafloor at 4,000 meters was disturbed 26 years ago, and not even the microbes had recovered.
No.
26 years later.
26 years in systems that have been developing
Millions.
tens and thousands and millions of
Yeah,
years.
yeah, yeah,
So when you say we've done a long time research at 26 years,
yeah,
I just have to laugh.
yeah, yeah. Well, yeah. I mean, we recently published some work where we went back to a 44-year. Is it 44? It's probably 46 or 47 years old now. Um, but with that one, yeah, there was some slight recovery in terms of some organisms, but other species just hadn't recovered.
Yeah.
You know,
And no, not
and
new, no new
sorry,
nodules.
what's
No
that?
new nodules.
No new
Except the
nodules,
nodules that
no,
are
no
there
new
are still
nodules.
growing.
I
Yeah.
mean, even the sediments, the sedimentation rates at these depths is about a millimeter or two per million years. So if you
Wow.
remove I'm sorry, a millimeter or two per thousand years. So if you remove 10 centimeters of mud when you collect these things, that's what's that? That's 100,000 years you've got to wait just for the sediment to
Blech.
you know,
Unreal.
resediment and for the profiles, the pore water profiles and the geochemical profiles to reestablish
To
themselves.
reestablish.
Yeah,
Really
it's
feels like our real focus should be, like on circular manufacturing and kind of making reuse of all the metals that we've already,
yeah,
mined.
but then we got to get on and do it,
Yeah,
I mean the amount
Solve
of
two
time
problems.
was
wouldn't it?
All the trash.
All the trash,
yeah,
all the landfills, all the dresser drawers full of old cell phones that people accumulate. What are we doing with all
yeah,
that kind of
yeah.
stuff?
Buy secondhand cell phones, buy secondhand computers.
Yeah, or
That's what we
figure out.
that's what I do with
Yeah,
my daughters, just they're not getting a new phone, they're getting a secondhand one. you know.
absolutely.
Um,
Well, we are creeping perilously close to the top of the hour, but I wanted to see if we could get through some of the questions that we have.
sure,
Oh,
yeah, yeah.
I'm gonna
Yeah,
keep this going.
I know.
I just got one more slide to show you, but
Okay.
we're actually coming across quite a few areas now where we're actually seeing localized increases in oxygen close to the seafloor in
Oh, that's
areas that
cool.
have no ventilation, um, in isolated basins where the salinity and the temperature they stay perfectly uniform with depth, so that there's no ventilation. And right as you get towards the seafloor, the oxygen starts to increase. So this may be not just a process happening on in the abyssal CCZ. This may be a lot more prevalent
That's,
than we think,
yeah,
which is really, really interesting.
it's
But this is
super
what we're trying to
interesting.
look at now over the next couple of years.
I'm thinking about the manganese crusts, right? That they talk about around some of the deep sea vents. So like maybe those are kind of doing similar thing.
Well, what's interesting is that if you go to some of these oxygen minimum zones where the oxygen can get down to about 2 micromoles per liter, it's hardly anything.
Hmm.
Uh, on ferromanganese crust, you can find higher faunal abundance and diversity and trophic diversity at the same depth band compared to other types of deposits like basalt rocks or whatever, which begs the question, A, where's the oxygen coming from?
Hmm.
And B, why are they only growing on these, or largely growing on these ferromanganese crusts? So yeah, there's lots of things to explore, and right now I'm just keeping a bit of an open mind.
Well, you should always do that, of course.
Yeah, yeah,
Be
yeah.
worried about preconceived ideas. Or
Yeah,
at least don't
Mhm.
don't fret if you have to let them go.
that's what we always say is just stay curious, right?
Yeah, yeah, yeah. What's
And
that quote by
be
Walt
prepared to be
Whitman?
surprised.
Be curious, not judgmental.
That's right. Be curious.
One of the things that it's attracted so much attention to your research on this is the level of oxygen loss, deoxygenation, if you will, owing to whatever it is we're doing to the ocean, what we're putting
Hmm.
into it, what we're taking out of it. We're disrupting ocean chemistry. which means
Mhm.
planetary chemistry.
Mhm.
So much wild animals. I wanted to ask if you think this is a purely physical kind of chemistry, or is it biologically anchored?
I, I, I mean, right now, I think a lot of these answers are found on page 971, and we're only on page 4 of the book. So a lot of these questions I have, is it biological in nature? Is it geological in nature? In our paper, we proposed a number of hypotheses, but they were just hypotheses, and and we said we hypothesized that this may have something to do with it. Um, and a lot of people took that as we think this is um, but yeah, so we just don't know. Is it biological? Is it abiological? Is it a mixture of both, or is it something completely different, like radioactivity induced splitting of water? I don't know. Um, Then we got to figure out, once we figure out what it is, how ecologically relevant it is. Is it just a transient process that happens when you have a bit of disturbance, or does this happen in lots of places, and ha has a wider ecological effect? For example, like on this screen, on this image here, where you're
Mm,
seeing
Mhm.
localized increases close to the seafloor. If that is oxygen production, then the oxygen production there is definitely having an effect on the ecosystem.
yeah.
Um, And then we've got to figure out how is it impacted by disturbance? You know, from whatever types of disturbance, deep sea fishing, deep sea mining, and do other types of habitats display this, this type of process? There's lots of studies now, for example, in groundwater caves and the oxygen minimum zone, the dark water column, where people are finding microbes and genes being expressed that
Mhm.
can produce oxygen in the dark. So I don't think this is anything new. I think we just found it in a different place.
Yeah, probably where you didn't expect to find it.
And but is it ecologically important? I don't know. I mean, it could be happening everywhere. We just don't measure it because the rates of respiration are higher than the rates of production.
Yeah,
In these chambers, you're measuring a net change. And if you have 10 grams of oxygen being produced, but 20 grams being consumed, you're not going to see any production.
Mhm.
Uh, so, but the fact that we find loads of microbes in these sediments, most of the microbes in these sediments, 30, 40% are able to generate oxygen. That's kind of
Kind of telling.
that's kind of telling.
so I recall that one of Carl Sagan's efforts to figure out how could you determine out there in space if there is life on a planet, water is essential, at least as far as we know, the way we um, think of life. But that detecting the presence of oxygen in the atmosphere was one clue that likely life must be there. That free oxygen in
Yeah,
the atmosphere is
yeah.
not considered to be um, abiotic, it's biotic.
Hmm.
Well, I mean, you can get, I think, on the surfaces of Titan and Europa, there's lots of oxygen, but it's radiolytic
Hmm.
in nature because the ice is basically bombarded by radiation from the sun, and then that
Physical
basically splits
process,
water.
Yeah.
Sorry?
physical, not
Yeah,
biological.
physical. Um, So yeah, I don't necessarily think that the finding oxygen is just an indication of life. I think it could mean something else. I was chatting to some NASA scientists about this. When was this? This would have been January, I think, right before I came out to
A
San
visit.
Francisco when,
Yeah.
when we met up. And they were saying, yeah, on Titan or Europa or on Enceladus, one of them, there was so much oxygen in the ice, and it was all because of, the water was being bombarded by radiation from the
So
sun,
it's
and
electrolysis of a
it's a
sort.
radiolysis.
Yeah, got it.
Radiolysis.
Splitting
Radiolysis.
Yeah,
hydrogen.
Yeah.
that's cool.
Yeah.
Cold, probably.
So
Anyway,
yeah,
Okay.
lots of different ways.
We we're gonna take a
okay.
short period of time and pop through the three open questions that we have.
Sure.
Neil is asking, what is the O2 concentration on the seafloor?
It's a great question. It really depends where you are.
Yeah.
So, for example, off the west coast of, well, the west coast, I was going to say the west coast of California. It only has a west coast. Off the coast of California, it's 11 o'clock here, so
We're sorry.
my brain's slowing down. But off the coast of California in the OMZ, I think the Davidson Seamount, the oxygen there gets down to about 2 micromoles per liter.
Wow.
Whereas in the Labrador Sea at the bottom, it's about 320, 330.
Whew,
So it
that's a lot.
varies.
Bless you.
Most of the oxygen, though, is most of the ocean floor is bathed in well-oxygenated water. If you go to the Black Sea, for example, there's no oxygen below 100 meters. It's completely anoxic.
Wow.
Yeah.
Excellent. Okay, we're going to go back to jellies for a minute. And Caroline is asking, what kind of jellies actually live in the deep sea? She said, we know that most get eaten by scavengers, but there are some that live there naturally. And she got to dive in Alvin some years
Oh,
ago.
excellent.
Yeah.
Excellent. Yeah. So in the purpose of full disclosure, I'm not a jellyfish researcher, so I don't know that many types of jellyfish. I was more interested in what happens when they die.
Yeah.
But one of the ones that we've worked a lot on in the deep sea is the crown jellyfish. They call it the crown jellyfish in Norway, paraphila, which has a cosmopolitan depth range. It's found down to 8,000 meters depth.
Wow.
But in Norway, in some of the fjords, the deep fjords, 400, 500 meters deep, they migrate up to the surface, and you can catch them with a net at night.
That's
And that's
cool.
how we were getting the jellyfish to then feed to the
Feed to the deep sea
critters.
critters. All right, last question. Neil is asking just for any final comments on the global effort to exploit the deep sea minerals, particularly around the CCZ.
Yeah, I mean, right now, in the CCZ, most of the license areas are being divvied up by the International Seabed Authority, which is still trying to ratify the mining code. I just heard that they weren't able to do it this past March. But there are some companies that are trying to mine now in national waters within the EEZ, deep sea nodules, and yeah, it's become a bit of a mess,
Yeah,
actually.
I would say so. And I'm curious whether the new high seas laws that have come into effect now will have any protective capacity
But
for that area.
you mean BBNJ and things
BBNJ,
like
yeah.
that. Yeah. I mean, I think all of these treaties have a role to play. Uh, I just think it's happening too fast, and
Agreed.
it's a bit tricky because lots of scientists, including myself, often say we don't know enough about the deep ocean to start mining it yet, but we can't keep using that argument, I don't think, because you can keep using that argument all
But
the
isn't the
time. Sorry.
precautionary principle
Yeah.
something we have to justify the real need? It's not about need, it's about money. It's about greed.
It is about money.
Exactly.
So
Yeah.
there's no
It isn't about need,
necessity
I don't think.
driving
Yeah.
the destruction of the deep sea in order to take something that is being sold as a product, and you're destroying a library. and
Yeah, yeah.
and a process that we're just beginning to ask the right questions, let alone have
Mhm.
answers.
Yeah.
Can you
Yeah,
talk?
and that's why we need a hell of a lot more time than
Yeah,
Six
but
months
it
or a
is
year.
in the future.
Yeah.
I'm
Hmm.
sorry?
Much more than 6 months or a year or whatever the latest
and
Oh yeah, and
yeah.
you need
realize
time to.
the
Sorry.
authority to make those decisions rests with about 30 people
Hmm.
representing the interests of 8 billion people,
Hmm.
plus all those who will
Yeah,
become
it's an
law.
important point.
Because
Yeah, yeah.
once you've done what you've done in the deep sea, that's it.
Yeah.
You can't
Hmm.
put it back together again.
No, no, it's not going to be possible, I think, to to restore it easily, especially the nodule fields. I think I did a calculation. It was a very, very rough calculation, but I think we calculated it would take something like 3,000 years of ship time to put the nodules back in one license area
Well,
Wow.
3,000 years doesn't
of
even
ship time.
begin to cut it. We're talking,
Well,
Add a couple
there's
zeros.
probably orders of
yeah,
magnitude differences, but yeah, it's happening too fast, I think,
Yeah.
and
Precautionary principle is the key.
yeah.
mhm.
Yeah.
I
All
think,
right. Well, we've kept you up too late there. I'm so sorry, but I was
Okay.
so
sorry,
grateful.
I'm so glad you came.
I'm so glad
Yeah.
too. So thank you, Andrew, and thank you, Ocean Elders, and thank you to everyone out there in the diving community who continues to show up to our webinars and to have these great conversations. We're so
Yeah.
grateful to everyone. Water connects us all. Remain curious, as we've been saying here, and spend some time out in nature. And we're going to be back again later this month. And until then, remember,
You have to protect the ocean
as if your life depends on it.
because it does.
It does. Thank you, Andrew.
No worries.
Thank you
Absolute pleasure.
so, really, so
Yeah.
very much.
Appreciate
And
the oxygen with every breath.
yeah, yeah. And yeah, have a good Easter break.
You too.
Yeah, yeah.
Bye for now.
Okay.